Hey everyone, we are going to look at your last lab kit lab today. So we are going to go into um, module 5 and click on lab kit labs. And this lab is all about biotechnology. So when you look at the lab manual, you will see that load in just a second. We're going to be looking at polymerase chain reactions as well as gel electrophoresis. These are both gel electrophoresis is kind of looking at like a DNA fingerprint, and then PCR is amplifying like small bits of DNA. So this kind of gives you some background information about sickle cell as well as what PCR is and how it's done. And then the steps of the cycle. This is just a figure kind of showing you, how PCR is done. And then you have a table here that will help you figure out the recognition site and the cleavage pattern. So this is the materials that you need for the kit or from the kit. And then these are the materials that you are going to have to supply, which is 3 to 5 9-volt batteries, microwave oven, everything else you should hopefully have at your house. But yeah, the 9-volt batteries, you need 3 to 5 of those. So what you will be doing in part 1 is essentially you're creating some electrodes. You're going to prepare some gel. This video will tell you exactly how to pour it. And you're going to be, when you're heating up the gel in the microwave, I know it says 30 seconds. Honestly, I would do 20 because I have seen this stuff explode very easily. So, and you don't want that all over your microwave. So you are done whenever the gel is completely melted. It shouldn't have any lumps in it at all. It's completely liquid. And then you're going to be pouring it into the chamber. You're going to let it cool, and then you're going to start doing the activity after the gel hardens. So that's going to take about 20 or 30 minutes. So activity 1 is looking at genotypes and determining if this child is going to inherit sickle cell. So there is some information that you're going to need to use from the table, and then you're going to essentially complete your gel electrophoresis. So you can see, like in this figure, it's kind of showing you the different samples, and then once you run the current through the gel, the parts of the DNA should separate out. There's a really good video here about loading the gel, as well as how to put the dyes in and doing everything, really carefully. This lab is kind of touchy, and hooking up the batteries so that way the current will go through. Don't connect more than 5. That's not gonna go well. So just be careful of that. And yeah, essentially that's it. If you look at your lab report, and again, in the class announcements, I did put some videos of like this lab kind of at play so that you can kind of see like what you're looking to do. So you have some questions to answer as you kind of walk through this. It's good to have this open while you're doing the lab itself. And then you're going to determine what is the genotype based on the information you get. And then this activity 2 is pretty short. You're just drawing a diagram include the dye that was put into the tray or the lanes, and then what bands appear once you run the electric current through it. So it shouldn't take too long. It's just complicated to kind of set up, but once you have it going, it shouldn't be a big deal. But let me know if you have any questions.